I'm excited to introduce you to Dr. Adrienne Godschalx. She brings a lot to our science team at Soil Food Web School, including her deep knowledge of chemical ecology and her phenomenal communication capacity. So, get ready to hear an amazing science story. Today, Dr. Godschalx will merge key ideas from her postdoctoral research in Switzerland with a vision for utilizing the soil food web to empower plant defenses. Adrienne spent years investigating the role of volatile plant compounds as signals to both microbes and insects. Now, she's bringing that knowledge to us so we can better manage plant and insect ecology, ultimately eliminating the need for toxic pesticides.
Hey everyone, great to see you again, this time as a speaker. Today I'm excited to talk to you about when is a pest not a pest. And I modeled this after a joke that I knew as a kiddo, like when is a car not a car, when it turns into a garage, something like that. So today, when is a pest not a pest? How soil food webs shaped plant-insect interactions. Again, my name is Dr. Adrienne Godschalx, and you can find me on the internet at the handle photosynthesoil. It's also my website name. And as you may know by now, I work for Dr. Elaine Ingham's Soil Food Web School as a mentor and researcher. To all my students out there, hey! But not only was this joke an artifact from my childhood, I also found some of my early literature. So, in 1998, fourth grade, I wrote, "I have a dream for the world, to save the rainforest, so the birds don't have to fly off to another place, and all the other animals would have a place to live, the trees and plants would have a place to grow." Apparently have been consistent throughout my entire career, and it all started with toucans. Even though growing up in the desert in Moab, Utah, it was beautiful there as well, I had wild dreams of getting to see a toucan. And so this video here is actually the very first, it's not technical, it's an asari, I never know how to pronounce them, but it is a toucan relative, but it was just this incredible interaction when I first saw this animal in person. So I thought I would share that with you as my starting point for how and why I'm here today talking to you about pesticides, because little did I know, but there was another woman who was brave and actively working out in the world at that very same time in 1998. But this woman actually had her PhD already and was actively publishing on how we can make soil more effectively useful so we don't have to cut down the rainforest. So I've now, long way around, gotten to a place where I feel like I'm working towards saving the rainforest. And that is thanks to this woman, Dr. Elaine Ingham. But more on her later. So let's get to the talk. When is a pest not a pest? So this starts with plant-insect interactions. What do I mean? Well, especially if you're a grower, you may have a few insects that come to mind. And I don't have time to represent every potential insect that you may consider a pest, but there's a few general groups. So we have first the sap-sucking insects. So these are insects that insert a stylet into the plant tissue, and actually it's the pressure of the phloem and the xylem that causes that liquid to move into the insect. They don't actually have to do a whole lot of sucking action because the plant has so much turgor pressure already, typically. So sap-sucking insects, aphids, whiteflies, etc. We also have leaf-mining insects. These are insects that will get between the leaf veins and get inside the leaf tissue, so they'll just eat around in that juicy, delicious pocket of food for them. We also have leaf-chewing insects, or as Elaine likes to say, comminutors, that essentially grind up everything they see and they just eat away. And of course, we have rasping insects, which I'm very fully aware that a snail is not an insect, but they have a radula, which does this rasping action. It's like a tractor, I don't know how to describe it. The radula moves back and forth and it scrapes, just like how thrips will eat and can cause quite a bit of damage. And of course, we also have plant-consuming insects below ground. In this case, we have many root-chewing larvae, which this is an example of the western corn rootworm, which is a beetle larvae. So it will grow up and become a beetle and then live above ground. But there's also other versions of this, wireworms, etc., that are typically younger life stages of other insects as they grow up and then move above ground. So, herbivores. Luckily, they're not the end of the story. We also have a crazy network of predators to keep their populations in check. Let's meet these predators. We have lacewings, which are also called aphid lions, which then, can you imagine what they eat? We have hoverfly larvae, as well as lady beetle larvae, which I want to point out that these hoverfly larvae can actually look like caterpillars and may not necessarily be recognizable. So if you see something that looks like a caterpillar, take a closer look and see if it's possibly a hoverfly larva, because they can be awesome at consuming aphids and other insects that could potentially outbreak. Also, lady beetle larvae. Many of you may know this, but this is what a lady beetle larva looks like that will grow up into being that lady beetle. I say lady beetle because they're technically beetles and not true bugs. And just so, in case you see these in the soil, I have students asking me about this from time to time, this brown feature is a pupated form of a moth or a butterfly, typically those leaf-chewing versions of the insects above ground. So after they finish chewing on the leaf, they will drop to the soil, form this pupa, and then come above ground with their beautiful butterfly wings, or moth, or however they choose to be in the world. We also have parasitoid wasps. There will be much more on them later. Predatory bugs. This is a minute pirate bug that is a nice predator for your thrips, if you have a thrips problem, as well as various sorts of beetles. Rove beetles are awesome predators. You may see these in your worm bin. Ground beetles are actually awesome omnivores that can take care of weed seeds in a field. So they're fun to have around, as well as they consume other insects as well. And of course, we have predators below ground, including entomopathogenic nematodes, and we'll talk more about them as well. And then, of course, we have higher trophic level predators, which I could no longer keep up with the arrows because it would just be a hot mess. But some of these may be classic, recognizable predators, but they're awesome to have in your field, depending on, again, who eats them and how the population dynamics are working. So we have praying mantises, we have dragonflies, and of course everyone has a parasitoid, including even the parasitoids. So there are parasitoids that can actually lay their eggs inside of the parasitoids that are inside of another insect. It's wild. Spiders are great to have around. Ants can be voracious predators and defend your plants, all depending on the interactions and the rest of the community. And of course, then we have the higher level cute predators that depend on these insect communities to be able to survive. Birds, fish, salamanders, and those large carnivores. So here's an ecosystem, or many. And of course, each of these represents groups and is not all inclusive, but gives you a picture for just how interconnected and how much depends on that first trophic level. So these herbivores really do link plants being able to photosynthesize and that becoming food, getting into the rest of this food web. So when we drop a pesticide, even if it's one targeted to a specific insect group, we knock out that link between plants as food to the next trophic level. So what may happen is that if predators consume some insects that have those pesticides, they can accumulate that inside their system and become even more toxic and knock out that predator population. Or, of course, predators looking for food when their main food source was killed off by a pesticide may not have any food left. And then these higher order predators are left without any food at all. So they really have no food, and then we get really sad fish die-off and other sorts of situations. So the situation can get pretty dire, but without predators around, certain insects are able to then reproduce because they no longer have their predators in the system. And certain insects, many insects, have the ability to evolve resistance to a certain pesticide. So then that insect that has evolved that resistance it no longer has its predator community and it can outbreak. So we have this increase in herbivore density on a plant, and this is what I will say is a pest. When we have an insect population that is out of control without a balancing predator community, we have a pest problem. But the typical solution in conventional terms in this dynamic has been historically to lay down some more pesticides. And I have this broken down into pest and icide because I want to point out that the term pesticide is, when we have a pest, here's something that can kill it, an icide. That's the root for to kill. So if we want to keep things alive, I say let's leave icides aside. Now bear with me. How can we do this? Well, in the great words of Dr. Vandana Shiva from last year's Soil Regen Summit, insects are not pests, especially when they're just serving as herbivores in this complex food web. And if we do not have pests, then we don't need pesticides. Great, we can all go home. But let me tell you a little bit more about how we can leave these icides aside. And I promise to do so using the soil food web. So let's look a little bit closer at these critters. So here's a graphical representation that I did of a flagellate in the upper side, testate amoeba, and a nematode. Now these microbes are very small. They're even so small that we can't see them with the naked eye, but we can grow them in our methods at the Soil Food Web School in aerobic composting methods, using the right ratio of components from our habitat, food waste, etc., high nitrogen, greens, browns. We can talk all about that. And then when we compost in an aerobic way, we can confirm that we have growth of the right food web organisms and the right balances using the microscope. And then we can then extract those organisms in a liquid form, making compost extracts and teas, and then further apply those out onto the field and cover your plant surfaces, your soil surfaces. And that is how we at the Soil Food Web School work with these microbes. So it's a lot of fun getting our hands dirty in the compost. And it's all thanks to this brilliant woman. I mentioned her before, but I'll give her another props because Elaine has really synthesized for me and my understanding of what I'm about to present to you in greater detail about how we can really interact with plants and insects by restoring this complete soil food web. She's connected geochemistry to plant physiology, to plant ecology, to this insect interaction world. So I really respect the work that she's done in putting together and synthesizing this framework that you're about to see a bit. And she has put together a model of the soil food web that she arranged in different trophic levels. You may have seen this in her talk. You will also see this, if you haven't seen it yet, in Dr. Edward Mitchell's talk. He found this as the first link for the soil food web. And benefits that we can get that Elaine tends to talk about are these 7 benefits that come from restoring a healthy soil food web. And today my talk is going to focus mostly on this one about protecting plants from diseases and pests. So, how do we do that without pesticides? This is the soil food web model for the most part. Elaine describes that we can have, with the soil food web, compete, consume, and inhibit. And I'm also going to add an additional one, which is induce. These are the principles at play that can allow a healthy soil food web to help us work with plant-insect interactions and prevent insects from becoming pests. So, the compete and inhibit are more directly connected to preventing against diseases. Consume and induce are more directly about pests, and that's where I'll spend more of my time today, but I'll cover all 4 of these. And to help you remember this as a little acronym, in Roman numerals, CCII comes out to be 202. And that, coincidentally, is also the area code if you wanted to call up your Congress folk and talk to your senators, talk to the folks in the U.S. if you're based in the U.S. And talk about how we can protect plants without pesticides. But I also learned in my journey, I got to go with Dr. Elaine Ingham and Jeffrey Smith and Dr. Tim LaSalle, and we got to talk with the Coalition for Regenerative Agriculture. And one of the big takeaways for me was that it's important to not just come in and talk about restricting pesticides, which I believe we should leave them aside, yes, but instead replacing with something that can be beneficial. So regenerative agriculture, as you're learning in all of these examples, can be an incredible replacement for this toxic chemistry that has been the norm. So instead of, I'm going to take a positive spin here and say, let's protect plants with soil microbes. And what does that mean? Let's get into it. So I found this beautiful scanning electron image of what really represents this first principle of compete. In the soil food web terms, we talk about this principle as the castle wall, that if a plant is totally covered with enough microbes, bacteria, fungi, nematodes, protozoa, then the pathogen, any pathogen that may land on that surface can't even find the plant itself. So if you get enough good coverage, that alone can be a defense against any kind of infection. So for no other reason than those exudates that the plant puts out, we'll talk about those later as well, are going to be swooped up by the organisms and not able to fuel metabolism for any kind of pest or pathogen organism. So how do they get there? Well, this is all about soil, isn't it? So as those organisms or those plants grow up out of the soil, they're going to be inoculated with it, which actually a study found when they were looking at how a Arabidopsis could be colonized by the soil microbes in a very protected chamber. They found that the soil bacteria could reach all of the leaves and flowers without any factor like wind, insects, or water. And to me, it makes sense. They're growing up out of the soil. What a better way to get inoculated. So it makes me then want to think, or even ask the question, how healthy is your soil that your plants are growing up through? If the soil itself is not already balanced, we have, like I said, we can make compost teas, we can make compost extracts, get those organisms back out onto those plant surfaces. And Dr. Elaine Ingham did some work that showed that when she covered grapevines with at least 70% coverage of a compost tea, it totally suppressed the botrytis that she also inoculated alongside. And then it was quantitatively, as she reduced the amount of coverage, the botrytis was able to take over and consume that leaf tissue more and more. So really just getting coverage is one of the best lines of defense. So compete is the castle wall. Consume. All right, here's where we start to think about these predators. So, unlikely friend may be ants. So plants can often produce things like extrafloral nectar, or what's famous in acacia, they have these domatia, these little houses, and they make these lipid bodies for the ants that actually live on that plant their entire life. Most ants don't have that kind of lifestyle with a plant, but they can often serve in this support of biocontrol. So when we say consume, we're talking about options to increase a certain organism that can control a population of potential pests. So ants really like to patrol plants that are covered in some sort of sugar, such as the plants that produce extrafloral nectar. They can also be effective defenders, as work that I did in collaboration with my PhD advisor, Dr. Daniel Ballhorn. The number of ants on a plant does negatively correlate with the number of herbivores. The beetle, in this case, would be herbivore on that plant. So you can see that the same plants had a lot of ants, had fewer beetles. But we also see that plants the same plants with ants and more parasitoids had fewer beetles. So this brings me to, what is a parasitoid? This is a parasitoid. It's not the aphid itself that looks like it's like hanging on for dear life. This is actually an aphid mummy. So these are awesome to see out in the field. And under a microscope, here's what it looks like. So this is, if you can see inside of that aphid body, there is the parasitoid wasp larva that is developing after it has consumed that aphid tissue, and eventually it cut a little hole for itself and popped out. And this is who that critter is and how they live. On a caterpillar, you can see here that the lady parasitoid wasp is laying her eggs on the top of that caterpillar, and then eventually that caterpillar became a caterpillar skin rug to keep these young developing parasitoid wasps nice and cozy. Another paper in the works. So parasitoid wasps actually can smell the signals coming from an herbivore chewing on the leaf, and one of the key players in figuring that out was Dr. Ted Turlings, who is also giving a talk, and I recommend listening to that one as well. So the plant puts out specific volatile cues. The wasp can smell it and follows it to the plant, and then, as you just saw, can provide some gruesome results for the herbivore. We also have the same story happening below ground, where herbivores, in this case the root-chewing larva, will release those volatile signals from the root tissue, and entomopathogenic nematodes can follow that to the herbivore and get inside of that herbivore, release a pathogenic bacterium that then preserves and makes that food. The whole full story is in Dr. Ted Turling's talk, but that's what it means to be entomopathogenic. So entomo meaning entomology, insect, pathogen meaning makes them sick or infected. Here is actually an image of a whole bunch of entomopathogenic nematodes that have just burst out of an insect larva cadaver, and we were collecting this for a project. It's pretty wild. This is through a dissecting microscope, so pretty fun to see all of them. One of the key researchers who first established this was my postdoc advisor, Dr. Sergio Rassman. He worked with Dr. Ted Turlings in his PhD, then went on to further study more about the soil interactions and how that can feed back and affect the above ground parts of the plant. And so this is one of our projects that I was able to work with Dr. Rassman and his master's student, Sandrine Fattore, and she was incredibly respectful with these organisms. I have massive respect for her and the amount of work that she did in this project. So I wanted to tell you about it a little bit. In this case, we put the entomopathogenic nematodes in that center sand chamber, and from there, the nematodes had free choice for which pot they would go to. And in this case, the question was, how would an earthworm in the soil affect the nematode's ability to find that herbivore and infect the herbivore? So the key finding there was that actually the turbation, the amount of passageways that the earthworm created, seems to have increased the diffusion rate of the key signal, beta-caryophyllene, that was able to make it through the soil pathways. So in this case, the earthworms actually support the entomopathogenic nematodes hunting ability. So there's interactive effects in the soil, and earthworms are important players, as we know, for many reasons, but also so that they can further enable this biocontrol system. So awesome. Great work, Sergio. Great work, Sandrine. So nematodes can also be a problem themselves, but luckily there are also predators of those other nematodes. Nematodes eat nematodes. Nematodes can eat everything. So the nematodes that eat your roots, there are predators for those to keep them under control. But there's also amoebae that can eat nematodes, or as you'll see in Dr. Edward Mitchell's talk, there's some pack hunting nematodes or amoebae that will surround and totally consume nematodes down to a nub. Here's a couple references for that. This is a great drawing of what these researchers found in 1995. And of course, there are fungi that also attack, trap, and consume the nematodes. So all of these organisms have been around for so long that they've all interacted together that this all makes sense, but it's pretty cool to see. So this beautiful image depicts actually three fungal cells surrounding the nematode, and then they swell up and trap the nematode inside of that little ring. But the fungus itself, as you can see on the left-hand side of this cycle, the fungus can switch into this trapping mode depending on certain cues. And I did a little bit of deep diving. This is all newer to me, but there are certain bacteria that when they're like, oh my gosh, we're being eaten by nematodes, they can actually trigger this nematode trapping fungus to start making traps by releasing certain forms of nitrogen. The key thing here too is also that fertilizers, abundance of nitrogen fertilizer, inhibits that switch over to nematode trapping. So if you have a root knot nematode problem, you might not want to overdo it with nitrogen fertilizers for several reasons, but that's one. Interesting, huh? You can also equip your plant to be ready for those nematodes before they even come, as this is an example of a paper that showed that you can colonize plant roots by these nematode trapping fungi, so they're already there inside, living inside of the plant roots, waiting for the nematodes, and able to be there as the first line of defense. So that's pretty cool. This was done with barley and tomato roots. And of course, fungi can also consume insects from the inside out, which is terrifying and beautiful all at the same time. This is a classic example of Bavaria. This can also be used for the fall armyworm and many other species combinations, but I wanted to point this out because these were fall armyworm photos of entomopathogenic fungi taken in Malawi. But the trick to getting biocontrol right when it comes to adding an organism in like this is understanding an insect's life cycle. So Cavi put out a really nice thing for the fall armyworm here that shows the insect's development on each different part of the plant, because it's really important to target the right parts of the plant at different times. This is a pretty devastating story because in 2016, the fall armyworm was introduced and has been devastating across Africa, and now into India and across Asia, down into Australia. Now it looks like it's in the South Pacific. Totally wild. And they're pretty aggressive herbivores that can come and just totally devastate a field. So I just want to point out also that Tieni is right in here and has been dealing with Malawi has been dealing with the fall armyworm now for some time. So I'm super curious to see how the deep bed farming and fall armyworm have interacted, if there's been additional benefits and protection because those plants have additional resources in those deep beds. So one way to also further support the consumption of herbivores is by putting in plants that specifically attract any kind of parasitoid wasp that could then kill off that herbivore, or specific plants that are very repulsive to specific herbivores as well. So it's called push-pull agriculture. And so I wanted to incorporate this one because it is from Malawi and thought it would be fun to engage in conversation about. But again, I'm so curious how we can incorporate push-pull agriculture, the Tieni deep bed method, and Dr. Elaine's soil food web models. All right, inhibit. So the main concept around inhibit is the production of antibiotics. And certain microbes are excellent, penicillin, for example, at making antibiotics. Or actinobacteria is another example. They make incredible, crazy fun compounds. Not fun if you're being the bacterium, but the antibiotics that they can produce are quite incredible. But the concept I want to emphasize more with this notion of inhibit is actually what happens when we have really good soil structure from a solid soil food web. And I'm not going to spend too much time because the benefits of fungi in soil are vast and complex, but one of the main ones is the ability to form macro aggregates. And so by forming these macro aggregates and introducing a really solid structure, we get incredible pore space, better water infiltration, and mainly oxygen. So water and air can infiltrate deeper into soil that has really awesome structure. You can see that more with DD's soil sponge. And in this case, the reason I have it here for protecting plants is because a lot of the organisms who can cause problems are often adapted to being able to live in these situations that fluctuate in oxygen conditions. So fully aerobic microorganisms tend to be beneficial for plant life, but a lot of the ones that can be pathogenic for us or for plants are adapted to living inside of a plant, inside of us, where there isn't quite as much oxygen flowing at all times. So inhibiting potential pathogens means let's keep it oxygenated. You know who else likes oxygen? Roots. Because plants themselves are aerobic microbes, so they need oxygen to live. So those root cells are constantly needing a certain level of oxygen. And so by having this pore structure that comes with having a complete soil food web, we get deeper and deeper root growth, which means more and more surface area, which takes us to my next plant protection method of induce. Now, to the plant chemical ecologists out there, inducing can have, it's kind of a loaded term because whenever a microbe interacts with a root, we can say that that induces a response above ground. But I'm going to hold this a little bit looser, and I'll explain why. So more roots equals more surface area, which means those plants get more and more nutrients. But let's think about this a little bit. How do plants get nutrients? I was taught in plant physiology that the nutrient acquisition is this diffusion gradient that plants put out organic acids that will kick off the protons off of those soil particles that are magnetically hanging out right by that clay particle, and then that will diffuse across to the root hair and feed the plant. Great, right? It all made sense except for, I think I missed a couple points on that test because I couldn't really fill in the gap between, but how does it really float across this space? I don't understand. Well, there's a whole lot of organisms in the way in a healthy soil, or even in an unhealthy soil, there may be a ton of bacteria there without additional organism groups. So if there are microbes covering all of those root and soil particle surfaces, what happens then? Well, it's not by accident. Plants are secreting tons and tons of exudates, and this is a dramatic image of a corn root that's secreting this mucus, and so it doesn't always look like, it's not always this like polysaccharide mucus, but also more of a liquid form of mostly sugars, partly carbs, and some proteins, as well as other secondary metabolites. But what Dr. Lane likes to share is, what if you went to the kitchen and you had made something with a recipe that was mostly sugar and carbs with some protein? Boom, we're making cookies and cakes. So if the plant is making cookies and cakes, there must be a reason. And actually, quite a bit of that plant's total photosynthate is being converted into sugars in the leaves. Photosynthesis, they put a lot of energy into that. And then that gets immediately pulled down into the roots and out into the soil so that they can recruit exactly the numbers and diversity of microbes that they want to have surrounding all of their root surfaces. So it can be up to 20 to 40%, or on average, it's 20 to 40% of a plant's photosynthate budget, but I've seen numbers of 70% or even 85% as well of the total plant's photosynthate budget, which is fairly expensive if you're a plant. So let's take this one step further. What's in a bacterium? And here's the raw data for you if you want to do the math yourself. But I will tell you that the C to N ratio of bacteria is 5:1. So 5 carbons for every 1 nitrogen. And this is how we're going to get to how nutrient really cycles. It doesn't magically float across the diffusion gradient. But if bacteria are 5 carbons for every nitrogen, and most predators are about 30 carbons for every nitrogen, the magic is in the stoichiometry, or the elemental math, basically. And how it works, and this is an example of one of these predators, a beautiful amoeba. If you want to see more examples of them, again, watch Dr. Edward Mitchell's talk. So if a predator eats 6 bacteria, because 6 times 5 is 30, they have to get 30 carbons for every nitrogen. So they're trying to get their 30 carbons. That also means that they will have consumed 6 nitrogens, but they only really needed 1 nitrogen for each of those 30 carbons in their own cellular metabolism, their own makeup of their body. Fun fact, we're also about 30:1 in general. And then the nitrogens in excess, because they have to get rid of that excess nitrogen or it becomes toxic, they have 5 nitrogens for every set of bacteria that they eat. So then we multiply that by the number of bacteria that they eat daily, which in even just this little GIF, there are a ton of little bacteria in there. So that's a lot of nitrogen that's being released by that predator. When protozoa were included, the nitrogen taken up by plants increased by 75%. And so the way this is working is that plants exude exudates, good vocabulary, and the microbes are specifically attracted to those compounds. They're colonizing that root surface. Predators come and munch, and then they poop out exactly the right proportion of plant available nitrogen that the plant needs to take up. It makes so much more sense. But they're not just made of nitrogen. What else is in a bacterium? We have all kinds of macro and micronutrients that is required for life, for membranes to function, for metabolism to happen. So what else is there? What else are those predators releasing at the plant surface? So let's go back even further one step to think about minerals. So this is my one geology slide for you. And I love these images because it's all graphical representation of how mineral particles contain nutrients. So in some of them, it's tiny little blue dots are the nutrients, or different colored dots between these magic carpets of silica layers. But in any case, we have essentially these ice cubes filled with glitter. If the glitter are all of the micro-macronutrients that a plant could ever want and need, they just can't access them themselves. The enzymes of bacteria and fungi come in, get what they need, put that into their own cells, and then they are ready to release that at the root surface when they get consumed. But do they have everything that a plant needs? This is the question. So I found this really neat paper that summarized all of the plant or the microbial metabolic pathways and what elements these bacteria are able to extract. And what it looks like is that all of the elements required for life are able to be extracted with their metabolic pathways. So if it's present in the glitter-filled ice cubes of the clay, soil, sand, silt, clay, rocks, pebbles, etc., then these microbes can extract it because they need it to survive. So microbial metabolism extracts all elements that are required for life. And luckily, a lot of the same things that a bacteria needs to live are the same things that a plant needs to live and thrive and be able to reproduce. So when we have a full balanced soil food web with predators able to extract and release those juicy bacteria and all of the contents within, then we get plant available micronutrients released right at the root surface. It's beautiful. If we take a few examples of micronutrients and their roles in plant and insect interactions, or in this case a pathogen as well, you can see that in the very bottom left corner of the first graph of A, there's a little dotted box, and that's indicating the concentration of zinc at which the fungal pathogen could no longer thrive. So the conclusion that this review paper is highlighting is that the pathogen has a lower optimal range of zinc than the plant. So plants with more zinc have more resistance. And this was true for other examples of pests as well, that the zinc toxicity can also be effective against insect herbivores. So when there's a complex soil food web with a diverse suite of microbes, including bacteria that can harvest enough zinc for their own metabolism, maybe even higher levels for specific microbes, specific species of bacteria, then plants are going to have access to that if there's enough predators to release that zinc at the root surface. Silica is another example here. So certain plants accumulate silica in their leaf tissues, oftentimes structure and toughness, in this case maize or corn. But that silica can also affect the plant's hormonal pathways. This case, the jasmonate pathway, which jasmonates interact with the salicylic acid pathway, which interacts with the ethylene pathway. It's this big, beautiful triangle of plant hormone interactions. And when the silica was present, it altered the way in which plant volatile compounds were released, and which compounds were released, which could affect the signaling pathway for that parasitoid wasp to be able to find their insect herbivore. But the availability of silica is actually dependent upon those soil parameters. And what do we know about the soil? We know that there are microbes out there that are using their enzymes to break down those rocks, pebbles, clay, and getting their micronutrients into the bacterial bodies. Those are being consumed by the predators and provided for the plant in appropriate concentrations. So taken together, after a couple examples, if microbes are providing a complete suite of macro and micronutrients, and I haven't even begun to tell you about how fungi can additionally contribute to nutrient availability, specifically the form of nitrogen, as the organic acids are going to modulate the pH surrounding those microhabitats, the microclimate pockets around different parts of the root surface that can affect the pH in a way that might inhibit the nitrifiers from taking the ammonium released by predators, which might otherwise get turned into nitrates. And that balance of nitrate to ammonium is a big feature in the balance of ecological succession. So we're not going to get into all of that, but the form of nitrogen has a big impact on plant chemistry, in addition to all of the other availability of nutrients that we just saw. So when I say induce, I mean the plant's resources, their nutrients, and how that induces their switchboards of choices that they make about which defense compounds to invest in and why. And the reason this matters is because the defensive compounds in a plant really determine what is edible and what is delicious. So here we have phenolics that you can quantitatively consume to certain levels, the cinnamon or the tannins. The wasabi and kales of the world have glucosinolates, a sulfur-based compound. All plants have terpenoids in them, big long terpenoids, but the shorter, more volatile terpenoids are highly abundant in certain plants, including mint and that lemon zest, pine trees, beautiful monoterpenes, sesquiterpenes. We also have alkaloids that can have some fun effects, spicy, caffeine, as well as my running joke that has lasted now for a decade, since 2012 when I started my dissertation work. If you need to, by chance, end the Hunger Games, because it's the chemistry of these berries, the fictional nightlock berries in the Hunger Games, that Katniss Everdeen threatens to consume that actually keeps her alive. So it all comes down to plant chemistry, whether one lives or dies, or in our case, what we enjoy or is delicious. But for a plant, What an insect really experiences is more like whether a defensive compound increases quantitatively in their system if they eat more of it. There's different varieties of these poisons that can kill them. Cyanide, sulfur-based glucosinolates, these alkaloids. Many defensive compounds are inducible. So as soon as a caterpillar starts consuming a leaf tissue, the plant actually recognizes the elicitors, those saliva components, which Dr. Ted Turlings also was the key player in discovering that. So feel free to ask him about those elicitors as well. But then the plant responds by knowing it's being consumed by an herbivore and cranks up its defenses throughout the plant. And of course, as we've already talked about, plants can make certain compounds that are not directly toxic to the herbivore themselves, but instead attract predators and make habitat more conducive to predators. So these are the herbivore choices that they're up against, which brings me back to our food web, because this first step in the food web all depends on plant chemistry. So plant chemistry is linking that all terrestrial food webs, whether it's a bison or an aphid or a caterpillar or a slug, the plant chemistry is going to determine how and how much of each plant food source to be able to consume. So the role of the soil food web has a major effect through this induction component on the plant above ground terrestrial food web. All right, let's take this together, summarize it all. Let's protect plants as soil microbes, competing using this castle wall, this full coverage, consume with biocontrol specific organisms that can attack and kill, inhibit using soil structure that can block out possibly organisms that have adapted to fluctuating oxygen conditions and have learned how to live pathogenically in a field, and by inducing, by providing a full balance of nutrition that can allow those plants to be equipped to not only have what they need to directly defend themselves, but to recruit help from their predator community. So in essence, I'm telling you that healthy soil food webs replace pesticides. We've got our microbes providing all of those nutrients, providing coverage, and the herbivores that in healthy balances can support populations of one another, they compete against one another and can help keep each other in check. Predators, which if there's enough of a food supply, can help be abundant in your field and control specific insects. And of course, our adorable higher level predators, including ourselves. You know, it's a good sign of the health of an ecosystem when there are large carnivores. And honestly, it's a really great sign when we are able to survive as well in relationship with this food web. Because when it all comes down to it, what we're relying on in this case is protecting plants with biodiversity. We need a diverse suite of bacteria to extract a diverse suite of micronutrients so that our plants have the full complex of ability to defend themselves. And that only comes with biodiversity all the way from the bottom, if bacteria are the bottom of the food web in this case, all the way up through our own metabolism. And because biodiversity is so fundamental, I want to take a moment and just express incredible gratitude to the indigenous populations of the world who are currently living in community and defending 80% of our global biodiversity, even though currently comprise less than 5% of the world's population and have been dispossessed of land across the world. So I want to just acknowledge that the existing communities and their ancestors since time immemorial have been advocating for these diverse communities and working with them and continue to today. So as a community today, what's our role? How do we help each other to continue defending this biodiversity that can enable us to even talk about regenerative agriculture and how we can use nature and work with nature instead of working with toxic pesticides? And I do believe this is the great work of our time. how can we work with nature, with one another in community? And like I like to say sometimes in webinars, if we want to work with fungi, we need to behave like fungi. So with that, I want to thank you. And if you have any further questions or you want to follow, connect with me, you can find me at photosynthesoil on Instagram and Twitter, or at my website, photosynthesoil.com. And I want to extend a deep gratitude for all of you for being here and being in community and wanting to use fewer pesticides and work with nature. Go soil, food web, microbes, woo!
Tieni means let's go. Tieni encourages women into leadership roles and provides essential farming inputs. For more videos like this, subscribe to our YouTube channel.